University of Delhi. I, Dr. Kamlesh Kumari, Assistant Professor in the Department of Zoology, welcome all the eminent speakers, guests from all the over the world and from the different walks of life. You have came here to share the knowledge and the vast experience with the science community. So here IMCD is designed to explore the scientific basis for the pharmaceutical and the nutraceutical for cancer and the other chronic diseases like your diabetes, obesity, mental health, arthritis, cardiovascular, pulmonary, infectious disease, and the rest can be uncovered under this platform. With this, I welcome our chairperson for this session, 3A, Emerging Anti-Cancerous Phytonutraceuticals 2. So we have two chairpersons for this session. The first is Professor Rupla. He is a senior scientist. He is a PhD, FNA, FNSC, FNASC, FMSI. His current affiliation is the Energy. He is a senior scientist in the Energy and the Resource Institute, New Delhi. His area of interest is molecular microbiology and the genomics and the metagenomics. He has done his PhD from the University of Delhi in 1975. He is the professor and the head of the department, Zoology Department from November 2017 to the 2010. He is the Dean Faculty of Sciences in the same University of Delhi. He is the chairperson of the Board of Research Society Studies, the member of Academic and the Executive Council in the same University of Delhi. From 2007 to 2010, he is the awardee of the Indo-US Professorship in 2008 to 9 by the American Society of the Microbiology (ASM) to conduct the research at the Oregon State University in the Corvallis. He is awarded with the Professor S.R. Vyas Memorial Award by the Association of Microbiologists of India. He is the recipient of the prestigious Mosilio Sachater Distinguished Services Award in 2016 from the American Society from Microbiology (ASM). We have another chairperson for the day, Professor Navneet Agnihotri. Ma'am is the professor and the head of the Department of Biochemistry, BMS Block 2, Punjab University, Chandigarh. Her area of interest is the nutrition and the cancer biology, lipid metabolites, reprogramming and cancer biology. She has done her PhD from the PGIMR, Chandigarh. She is rewarded with the ICMR International Fellowship for the Young Scientists in 2012 to work at MD Anderson's Cancer Center, Texas. She is awarded with the National Scholarship by the Chandigarh administration in 1986 to 87, and she is also awarded fellowship by the PGIMR for the PhD in 1988. We all welcome you, ma'am and sir, for this session. Now I request the chairperson to start over the sessions.
So it's a pleasure to introduce Professor KL6 Kundu. Probably all of you know that he is now currently at the Kalinga Institute of Industrial Technology, Institute of Happiness, Bhubaneswar, and you are again familiar with his work in tumor biology, cancer, stem cells, tumors, trauma interaction, and so on and so forth. He did his PhD from Bose Institute, Kolkata, had several postdoctoral fellowships primarily in USA. He was the Dean and Director at NCCS Pune, Professor of Medicine and at KIT at School of Biotechnology, Bhubaneswar, has more than 116 research publications, one US patent, and he is the Shanti Swarup Bhatnagar Awardee. And probably it is a pleasure today to listen to him. Many times when I talk to Mrityunjay, he always starts the discussion with your researches and everything. So we are familiar, and it will be a pleasure for us to hear you today, sir. Thank you.
Good morning. I'd like to thank Professor Alok, Professor Ajay, Professor Subhas to organize this event. I remember I also attended that earlier INCD, one organized in Rishikesh and another one at IIT Guwahati. Of course, I need to thank Professor B.B. Agarwal, who I knew my first interaction with him back in 2003 when I published 5 papers in 1 year, Journal of Biological Chemistry from National Center for Cell Science. One day I got a mail from Professor B.B. Agarwal that Dr. Kundu, I saw your paper. I need to get one cDNA construct called LCK, leukocyte kinase. I did not know who was Professor Agarwal. Then I sent it from Pune by post, and then immediately he contacted. He said thank you. He was trying from other university in US, but when he saw our paper, and then that was my first interaction. And since 2003, then I was part of his family, Society of Translational Cancer Research, INCD. I was also co-organizer of his 3rd International Conference on Society of Translational Cancer Research, which we organized in Lonavala, I think back in 2007, if I remember. With that, I will not take much time because we started late, and also I'll try to finish within 20 minutes. So after coming back India, staying almost 9-10 years, including NIH, so I never worked in the area of cancer when I spent 10 years in US, especially work on cardiovascular research and other area with chemistry background, moving to molecular biology, cell biology, immunology, etc. But after coming back India, that time director said that you have to start something new. So that was my beginning of cancer research journey. So title, as you can see, therapeutic targeting of different subtypes of breast cancer using nutraceutical based approach. I'll just share some information. This is very unique, and this is how it happened in India. So when I was director in CCS, short span of time, and so I submitted one project along with my team to DBT BIRAC as well as funded by World Bank through National Biopharma Mission Program. It's called NBM. And that objective of that program was establishment of GMP compliant national repository for banking, safe deposit and supply of characterized cells for use in biopharma industry in India. There is no government program so far on this area. So we submitted, I was coordinator of the project, and after multiple review and multiple interaction, project was funded 24.5 crores INR for 5 years. And the purpose of that was that biopharma industry in India, those who are developing druggable molecules, they have to independently test in other facility. Mostly they are test outside India, which is very expensive. So the government decided that can we have that kind of, such kind of facility in India. But unfortunately, I got this project 2019 beginning, and 2019 November I had to leave NCCS Pune, and after that next director could not activate this project, although they received 50% of fund, about 15 crores they received. Till today the project is not activated. Coming to cancer, many of you know, and then yesterday there is a lot of talk. So the message I like to give, I don't see many students here, although Alok spent huge time and efforts and fund to organize this conference. I was expecting more students would participate in this program. So any nutraceuticals or phytopharmaceuticals, if you want to test it, you need to have a good validation, scientific validation, good target, good model. And today, any cancer, any solid cancer, unless until you understand that tumor heterogeneity, you understand the tumor microenvironment, it is very difficult to know where you are targeting, whether you are targeting in the tumor cells or you are targeting in the stromal cells, which includes cancer-associated fibroblasts, endothelial cells, many immune cells, including macrophages, tumor-activated macrophage, NK cells, T cells, pericytes, and many other cells, including some subsets of cells that is called tumor-initiating cells, TIC, or cancer stem cell. So I'll be talking mostly breast cancer, but I will show some data related to other cancer as well. So breast cancer, those of you who are working, in general 4 subtypes: luminal A, where the ERP are positive, HER2 negative, luminal B, all 3 positive, hormone receptor, HER2 overexpressing, 4th one TNBC or basal line. What I have seen, I will show you slides with my other collaboration. India now, earlier cervical cancer was the #1, but last 5, 6, 7 years, among women breast cancer becoming #1. And the patients, what I can see with my collaborators lab, Tata Memorial Hospital, Mumbai, as well as my current cancer center, Kolinko Cancer Center, KCC, KIMS, KIT University. Women age group of even before marriage, age group of 27, 29, 30, 31, they are developing triple negative breast cancer, very aggressive, unlike luminal A, luminal B or HER2 positive breast cancer. The last one, that is called IBC, inflammatory breast cancer, not very common in India, where the entire organ become inflamed. This is how it works. Starts with the normal duct, when it becomes carcinogenic, and then this is called ductal carcinoma, and when these cancer cells become more invasive and aggressive, that is called invasive ductal carcinoma, and it happens in that lobule also, that is called lobular carcinoma. Started with a little lump here and there, as you can see, and then tumor grow at the primary site bigger, and initially targets in the lymph node. Lymphatic area is the first target. From there it goes to lung, liver, and bone and brain, like prostate cancer as well. So therefore, understanding that development of the cancer at the primary site as well as metastatic site, especially need to do a lot of work in order to understand that mechanism and progression of metastasis. This is one of the projects we are running. Still this project going on, 5 years project, when Professor Harold Verma visited, when Dr. M.K. Van was Secretary Deputy, he is no more. That time he suggested, can India have NCI India, which started in Jhajjar with 3 components, and parallelly DBT can have a VNCI program, DBT VNCI, Virtual National Cancer Institute program, in all different cancer. It's like a NCI spore kind of program. So I was part of that breast cancer VNCI, started around 2017 with 5 institutes: Tata Memorial Hospital, TMH, ACTREC, National Institute of Biomedical Genetics, NIBMG, IOB, Professor Akhilesh Pandey, part of that, and but he moved from Johns Hopkins to Mayo Clinic, Rochester now. So he was not able to continue this project, and I started from NCCS and transferred this project to KIT University. So objective of these projects were established primary culture and cell line from hormone resistance and sensitive breast cancer patients tissue, and all samples with proper IRB and ethical issue, ethical consent and clearance sample will be collecting from Tata Memorial Hospital, and that's the way we have done it. Second component was my component along with first component. development of patient-derived xenograft, PDX model. This is unique model. Many countries they have developed all different kind of cancers and their respective PDX model, but we are always late. So India, I don't see any PDX model so far. Once you develop PDX model, screening of new drugs, testing efficacy of selective known drugs, identifying therapeutically relevant biomarkers in this PDX model. Number 3, identification, validation of novel therapeutic target from either directly from patient or PDX or primary culture using phosphoproteomics, whole genome sequencing, whole exome sequencing and transcriptome based approach. So this was the collaborative project, and my role was that to collect sample from Tata Memorial Hospital. I started this when I was at NCCS, and now this project continuing one more year till 2023. So depending on the size of the sample, some sample we directly implant into the immune deficient mice to develop PDX. This is very time consuming because PDX takes more time than injecting cancer cells. Then you have to generate generation 1 to generation 2, 3, 4, etc. to see that they are consistent, they are showing the same phenotype and same pattern what you see in the patients. If they are luminal positive, they should show that luminal positivity in that generation 3, 4, 5, etc. Some of that we directly processed for establishing primary culture, thereafter establishing cell line, because till today we don't have any India-centric breast cancer cell line available. Depending on the size of that tissue, some of that we process for both PDX and primary culture. These are the sample identification number, patient's ID number. As I said, some used for primary culture and some used for PDX and some combination of both. These are the students left and right, they work in this project, and representative tumor. Since these are luminal positive, so we had to check the status of ER alpha, PR alpha, R2, along with EMT specific and proliferation specific markers such as ECAD, NCAD, Vimentin, Chi67, pan-cytokeratin, etc. So this is I am showing generation 1, patient ID number P014R. Patients admitted Tata Memorial Hospital around 2015-16 with multiple treatment, combination of treatment, and then patient passed away I think in 2020 or 2021, but we got all the samples. And these are again some representative of different patient-specific PDX, and these are generation 4. As you can see, left side panel A, the representative tumor, panel B, immunohistochemistry, immunofluorescence and confocal data, along with some other signatures and marker, including alpha SMA. They are showing ER positive, they are showing PR positive, that is the final message, R2 positive, and some of that Western data, panel C. So then we did some differential expression of this gene, comparing patients versus G1, G2. And we found that there are multiple pathways specific, including that cell signaling pathways, MAP kinase pathways, apoptosis pathway specific, metastasis pathway specific genes, they are expressing in these sets of data. Panel A, we are showing representative tumor from generation 1 to generation 5, along with that immunofluorescence and confocal. Panel B, showing that palpability of the tumor and the growth of the tumor. If you can see with the higher generation, time taken to grow the tumor and to become palpable is shorter than that generation, earlier generation. And we have done that correlation of gene expression by gene spring, panel C and panel D. And what we can see, they are tightly correlated between that patients as well as generation 1 and higher generation. And based on gene ontology analysis also, we have checked that functional annotation of genes which are differentially expressed between primary tumor and different generation of PDX. So I'll now go to that some other area. We also did some work related to curcumin. Earlier days, we have shown that curcumin not only targeting NF-kappa B, it target also that one of the proteins or genes which work on osteopontin. So it target osteopontin regulated angiogenesis by specifically targeting these transcription factor, including NF-kappa B. It came earlier cover page of the journal. Recently, 2019, after moving to KIT University, I got one more project through DBT related to curcumin, nanocurcumin formulation, along with drugs like 5-fluorouracil and other drugs targeting breast cancer. So this was like multi-pronged approach for preclinical assessment of novel curcumin nanoformulation as a drug for breast cancer therapy, and testing in that patient-derived primary culture PDX model to check their acute and chronic toxicity, their pharmacokinetic study, biocompatibility, drug release, long-term stability. So this is also under mission mode project because DBT started that independent curcumin task force 3-4 years back with Professor G. Padmanabhan as a chairman. So we submitted this project along with my clinicians from KIMS, KIT. We got about 1 crore project. So we generate a lot of data. Maybe next INCD I will present this data. Another phytopharmaceuticals related, yesterday other people talked about this androcaphilite. In collaboration with the National Chemical Laboratory, NCL Pune, Dr. Tulsi Rao, who is basically by training chemist, he purified this and characterized by mass spec, HPLC, etc. And then we have tested in our breast cancer model, and we find that androcaphilite suppressing breast cancer growth. As you can see, panel C, D, tumor growth, tumor volume, tumor weight. And this has been done using that breast cancer cells tagging with the luciferase genes, because this is the bioluminescence technology with that ID spectrum model. We have captured this image and quantified this image. And I was the first person in CCS to develop this technology, going to Hokkaido University, Japan for 1 month as a visiting professor, learned this technology, came back, and 2009-10 I started this technology in India first time. I will switch a little bit to skin cancer, connecting with this androcaphilate story. Many of you know skin cancer is a major issue, especially Europe, Australia, US, mostly because of mutation of BRAF and NRAS, and major cases patient die because of lung metastasis. One of my students, Hiraj, he did PhD, then moved to MD Anderson Cancer Center, and then moved to Columbia University. His boss moved to Columbia University. Now he joined as a group leader at Johnson & Johnson. He asked this question that whether cancer stem cell exists in skin cancer model. So we started with that all melanoma skin cancer specific cell, starting with B16, F10, whatever available, mouse specific, F1, human specific, A375, SKML2, 28, with different mutation of BRAF or NRAS, and whatever signatures or marker available to characterize cancer stem cell, we use almost all in this particular model, starting with CD133, 20, 271, ABCG2, VEGF-R1, CD166, CD44, etc. We thought, can we have a double positive cancer stem cell? But when we did that by FACS sorting, we find that they are less than 1%, 0.8%. So it's very difficult to, as a graduate student, PhD student, to continue the project because it's very laborious work. So then we decided to work on CD133 and positive and negative cancer stem cell, and move further and to check first what are the differential express genes which are present in CD133 positive versus negative, heat map analysis, volcano plots, and then I am not showing all details data because this conference is related to nutraceuticals. So then we developed that good lung metastasis model, and then we tested this multiple by, so we developed lung metastasis model by injecting CD133 positive cancer stem cell and looking at multiple druggable molecule including andrographite, which somebody shown in that clinical trial going on in that colorectal cancer CRC. So we also tested that. So this is our B16F10 LUC derived CD133 positive cancer stem cell injected through tail vein, develop lung mets model. And this is X5O image you can see. Left one is CD133 positive, right side CD133 negative cancer stem cell. Panel B, you can see that micro and macro mets. And panel C, the level of VEGF expression also gone down in response to andrographite with 150 mg per kg body weight. Again, the issue here, this paper published Journal of Investigative Dermatology, which is one of the top journal in the area of dermatology. But we extensively dissected that mechanism, how that CD133 positive cancer stem cell develop lung metastasis. We find that Notch1 is predominantly active in CD133 positive cancer stem cell. We also find that andrographite behaving like a gamma secretase inhibitor like GSI, which blocks that to formation of active notch called NICD1 from Notch1, and then NICD1 ultimately suppress that CD133 dependent MAP kinase activation including P38 MAPK, and that inhibits that transcription factor AP1 dependent MMP29 and BCL expression, and thereafter suppression of angiogenesis and metastasis. We have also used multiple drugs like tramitinib, dacarbazine, dabrafenib. Those are the three drugs today using for treatment of skin cancer. In combination of drugs or independently with their IC50 etc., we checked with GSI and also checked with andrographite, but andrographite also suppressing lung metastasis. As I said, this paper published Journal of Investigative Dermatology. Now I'll talk a little bit about, I'll finish maybe within next 5 minutes. This is one of the protein which we working last more than 22 years NCCS and last 3 years KIT University. This is basically chemokine-like protein. This protein interacts with integrin. Some of you will be knowing that this year Lasker Award received by Professor Richard Hines and Eric Bruslauti based on their major contribution on discovery of integrins. There are many heterodimers, and osteopontin is also one of that ligand interact with alpha V beta 3 integrin as well as some other integrin depending on the nature and type of cells. So there are huge international community, they work in this area. There is International Osteopontin Society, and this protein basically has a unique RGD motif, GRGDS, thrombin cleavage site. Basically this protein once interact with either integrin or CD44, it regulates cell-cell communication, cell matrix interactions, angiogenesis, cell addition, migration, etc. And as I said, internationally there are many groups, Neil Federico from Johns Hopkins, Gary Fisher from NIDC or NIH, Bob Weinberg, MIT Harvard, and many other groups in Europe, Japan, Australia work on this. Canada work on this area. European group some couple of years back, circulating osteopontin act as a predictive marker in hepatocellular cancer. They studied 350 patients One of my students completed PhD with me, moved to Yale University, had a paper in cancer cell, shown that osteopontin CD44 axis in row GD12 mediated metastasis suppression. Similarly, osteopontin CD44 also promotes CSC, cancer stem cell phenotype in glioma. Somebody published in Cell Stem Cell. These are very high impact journals. Bob Weinberg published Cell 2008. I'll just come to that paper. As I said, they started with Gordon Research Conference, then Facet Research, every alternative year, and now we have our own international society. From India, I am the only representative. You can see my name there also. This was 2017 conference in Renaissance, Baltimore, Harbor Hotel, Baltimore. But we do this conference across US. Last 2 years we could not do it, but again hopefully next year we will be doing. Many guys, including Professor Harvey Kenter, very well-known immunologist from Dana-Farber Cancer Center. As I said, Professor Neil Federico, then Professor Susan Ridlings, George Weber, and many, many others. Amanda Brown from Johns Hopkins, etc. So, targeting osteopontin, and I know my earlier journey, and Professor Agarwal, I was following his series of papers, Cancer Research, JBC, many, many other journals, Clinical Cancer Research. So, what he was doing, that how TNF-alpha regulates NF-kappa B and MMP's expression, activation, and with a series of active compounds from varieties of species, spice. And what we have shown that how osteopontin controls tumor growth, metastasis, and angiogenesis by selectively targeting some of the specific genes which are pro-angiogenic in nature, like cyclooxygenase 2, VEGF, some of the other targets like P38 MAPK. Today, if you search it, P38 MAPK is one of the hotspots in triple-negative breast cancer. So you can see that from India, we published series of papers in cancer research back to back, all targeting this area. Bob Weinberg, I told you the paper, what he published, systemic endocrine instigation of indolent tumor growth requires osteopontin. I am not going into details of this. Final message of this paper is shown that blocking osteopontin's action may yield useful cancer treatment. And so the first author of this paper, Sandra McKester, she is now assistant professor of MIT Harvard. And I had chance to meet Professor Weinberg 2016. And since then he is sharing some of his reagent from his lab, published large number of papers. He is that pioneer of hallmark of cancer, 2 Cell paper 2000 and 2011. Again, that osteopontin also, other nomenclature called small intriguin binding ligand L-linked glycoprotein, sibling. This is the nomenclature given by Professor Neil Federico, Larry Fisher, etc. Basically, osteopontin and other family member of this protein, they call CCN family, like BSP, DMP1. They binds with integrin, but they regulates starting from survival to apoptosis, proliferation, invasion, angiogenesis, metastasis. They published paper in Nature Review Cancer, one of the review article. I was reviewer of this Nature Review Cancer also. Now I'll just show you, yesterday Professor Paul was talking something about that breast cancer model, prostate cancer model. So here what we have done recently, this is very recent studies, how osteopontin transactivates normal fibroblast to myofibroblast or cancer associated fibroblast, and how that controls breast cancer progression. This is review article by Raghu Kaluri from MD Anderson, Nature Reviews. So you know there are many genes including extracellular matrix protease, chemokines, growth factors. they are involved in the differentiation of fibroblast left side to myofibroblast or activated fibroblast or cancer associated fibroblast called CAF or TAF. And once they differentiate, many specific genes like stroma derived factor 1, alpha SMA, SDF, FSP, they start to express. So one of my students, Ramesh, he asked the question whether osteopontin, being a chemokine-like protein, ECM protein, whether regulates this or not. So we started with the human fibroblast cells and we isolated peripheral normal fibroblast from breast cancer patient and then exposed with osteopontin, find that yes, it differentiates fibroblast to myofibroblast. Again, I am not, because of shortage of time, I am not going through details, but we find that osteopontin, irrespective of interacting with alpha V beta 3 integrin or CD44, it regulates transcription factor called twist1. Twist1 is one of the transcription factor which is highly activated and expressed in cancer associated fibroblast along with NF-kappa B and AP1 through ERK and AKT pathway, and that ultimately controls alpha SMA, FAP, FSP, especially SDF and alpha SMA, which we have shown by reporter promoter assay, by CHIP assay, by Western, qPCR, etc. And we have developed two independent tumor model, one carcinogen induced tumor model, another orthotopic organotropic tube breast cancer model. By silencing with lenti-based shRNA, I am not showing data. This twist1 etc. also regulates microRNA MID21, which ultimately controls, there is a feedback AKT through P10 being a tumor suppressor. And this paper recently published on Oncogene 2021. Ramesh moved to University of Texas Southwestern, Dallas. He started his postdoc. So in this series also now, last 5-6 years, we had series of paper in Oncogene. Starting 2013, 14, 15, 16, and 18 one more and 21. And again that recently we are using some of the CRISPR-Cas9 based approach. Recently paper published in Molecular Cancer is a review article, impact factor 41. CRISPR based therapeutics, a new paradigm in cancer precision medicine, along with some of that paper in Cancer Cell International, who are targeting that immune cells in the tumor, how that IL-6 regulates that STAT3 in that macrophage in the breast tumor model, and some other paper. So therefore the take home message, osteopontin is one of that important therapeutic target, and we have published series of review article including expert opinion therapeutic target, 3 or 4 issues, and 10 cell biology and some others. We are searching for small molecule inhibitor to block the function of osteopontin, because we have done extensive mechanistic studies. Now we are looking for many nutraceuticals and in combination with drugs and with a good model. Good model is very important. If you don't have good model, either preclinical animal model or organoid model, 2D, 3D model, is very difficult to validate. So we find that osteopontin also targeting a androgapillide targeting osteopontin, it blocks that expression of osteopontin. We are also using some of that siRNA, shRNA targeting osteopontin, neutralizing blocking antibody targeting osteopontin, alpha V beta 3 integrin CD44 in collaboration with Murdoch University, Australia. We are using that aftermath technology, basically targeting tumor endothelial interactions, angiogenesis, metastasis and tumor growth at the primary site. Last few slides. With nanomedicine based approach, all of you know that there are many drugs in the market, nanomedicine specific drugs. One of the best example is Abraxane, which is albumin tag paclitaxel. It is discovered by Abraxan, US-based company, Abrax, US-based company, and now Biocon and many others, they are making the generic form of this Abraxan. And with that approach, last 10-15 years we are collaborating with IIT Bombay and many others. We are using chitosan as a biocompatible, biodegradable US FDA approved nanoparticle. With my chemistry background, we do a lot of conjugation chemistry. Here we have done with that SPDP with the cyclic GRGDS peptide conjugated with chitosan and then tagged with androgaphyllite or curcumin or other drugs in combination. Here I am showing that androgaphyllite nano formulation, their size, zeta potential, cellular uptake, and we are using raloxifene as a control, raloxifene nanoformulation versus androgaphyllite nanoformulation. Both of them independently suppressing tumor growth in breast cancer model, 41 and other cancer model. Their toxicity, hematological, biochemical, they are not toxic in healthy mice. We have shown that, and we have patented. Last slide, some of the translational approach based on our 25 years journey in India, using some anti-PD1, PDL1 along with anti-osteopontin as a targeted immune therapy in triple-negative breast cancer. Second approach, drugs like raloxifene and other drugs along with nano formulation targeting in TNBC. Third one, Notch and osteopontin dual inhibitor along with anti-HER2 or anti-estrogen therapy in luminal AB and HER2 positive breast cancer. Fourth one, mTOR and PI3K dual inhibitor along with COX2. Many of you know that like aspirin and COX2 inhibitor now, they are working very well in different cancers through repurposing of drugs mode. So we are using mTOR PI3K along with COX2 inhibitor in our model. And the fourth one is melanoma skin cancer. We are using that dabrafenib, tramitinib along with Notch or PM osteopontin dual inhibitor in melanoma model. As I told you, we are using organoid, soft agar, transwell, mammosphere, melanosphere acids, 3D model. We are also using organotropic, orthotopic PDX model. And now through outsourcing, we are studying the drug release, toxicity, PK, PD, ADME, etc. with some selected formulation. With that, 23 students completed PhD degree, majority of them settled in US, many of them now assistant associate professor. Currently my second innings at KIT, 5 PhD students including couple of students still there at NCCS, 2 postdoc, 3 project student, running 6 equivalent to NIH R01 project including Indo-German. Many collaboration with University Oslo, Hannover Medical Center, Murdoch University, Curtin University, Louisiana State University, School of Medicine, IIT Bombay, etc. Some of the students here, some other students. Thank you very much.
Thank you, Professor Kundu, for your excellent talk on breast cancer, microenvironment, skin cancer, and also some nano drugs and nano formulations. We are running out of time, so probably I know you, all of you must be having a lot of questions, but you can interact with Professor Kundu after the session, and